We will spend the next 20 minutes or so exploring gene regulation in eukaryotes. Some of the main topics we'll focus on are listed on the screen there. And let's begin with making some comparisons with gene regulation in prokaryotes. So when we think about gene regulation in eukaryotes, what we're going to see is that it's far more complicated than that in prokaryotes. Why is this the case? Well, just think about it. Eukaryotes, including multicellular organisms such as ourselves, are complex. We have complicated cells with lots of different organelles. We have lots of different types of tissues and organs, each of which might have distinct functions. And we go through complex developmental stages over extended periods of time. So again, these are all radically different than what happens during the life of your typical prokaryote, such as bacteria or archaea. So if we make some general comparisons between prokaryotes and eukaryotes, what we're looking at here are just some highlights in terms of differences in gene regulation in prokaryotes and eukaryotes. So if we imagine prokaryotes, such as E. coli or most other bacteria, often we find genes organized in operons, and they might be under the control of a single promoter. However, in eukaryotes, what we tend to see is each gene is separately controlled by a single promoter. In prokaryotes, you tend to have just one or maybe a couple proteins that can serve as activators or repressors. But in eukaryotes, what we're going to see is that lots and lots of proteins, various transcription factors, have to come together in order to regulate gene expression. In prokaryotes, we might have just one or maybe two DNA sequences that serve as switches, such as an operator region, for example. And they tend to be located just upstream of structural genes. However, in eukaryotes, we're going to see that there are multiple DNA regions that can serve as switches that can play a big role in regulating whether or not genes are expressed. And finally, in prokaryotes, for the most part, there certainly are exceptions, but for the most part, the ground state of most genes is on. In other words, genes tend to be expressed unless there's reason for them to be suppressed. In eukaryotes, on the other hand, it's sort of the opposite. By and large, most of our genes in most of our cells tend not to be expressed, but they are only expressed when they need to be, and they're under various activation controls. We'll talk a bit later about this concept of chromatin and how DNA interacts with lots of different proteins to form chromatin within our cells. This is a figure from your book illustrating some of the basic differences between gene expression, again, in prokaryotes and eukaryotes. So toward the left, again, just shown graphically here, we might have a single operator region. Here we have a single RNA polymerase binding to the promoter area, and that would then make its way down the DNA, ultimately transcribing whatever genes we're discussing here. Again, the point here is in both prokaryotes, by default, most genes tend to be on or expressed most of the time. Eukaryotes, however, toward the right, different situation. Our DNA, shown in light blue here, typically is wrapped around lots of proteins, shown here in purple. And for the most part, most of the DNA and the genes located in the DNA, they're not available for expression. So again, for the most part, most of our genes tend to be in an off state, unless other proteins come along and activate their expression. Just to give you a sense of some of the complexity regarding the organization of coding regions and regulatory regions in eukaryotes, if you take a look at this schematic here, again, we're looking at just a small portion of DNA from some eukaryote. If you focus initially on the coding region, illustrated by the introns and exons right here, notice in addition to the coding region, there are lots of regulatory regions that play a big role in whether or not this gene is expressed. So for example, just upstream, here's a promoter region right there. There's this region here called the Promoter-Proximal element. There are these other regions referred to as enhancers. They might be way upstream, like we see here. Way downstream, they might even be found within introns. In addition, although they're non-labeled here, there are other regions referred to as silencers, which they play a role in inhibiting or repressing gene expression. So the point is it's pretty complex, and there are lots of different DNA regions that play a role in whether or not a particular gene will be expressed. If we take a few seconds and look at some of these regulatory regions, these regulatory elements in eukaryotic DNA, just a few comments here. So first of all, the promoter region. This is characterized by having this very characteristic TA sequence, so-called Ta-Ta box. Again, this is characteristic of promoters of essentially all genes in eukaryotes. Just a few comments about the promoter-proximal elements. Again, they tend to be just upstream, but very close to the promoter. Common examples are the so-called CAT box and the GC-rich box, like we see illustrated here. Again, these represent regions that, as we'll see in a bit, where certain proteins that serve as transcription factors have to bind to in order for gene expression to occur. So in other words, in order for the coding region of this particular gene to be transcribed to produce mRNA, such as we see toward the far right of the diagram. Just a few comments about some of the evidence behind the promoter region and these promoter-proximal element regions. So what we're looking at here is some data whereby we're just simply illustrating if you mutate a lot of the bases near the core promoter, like the TATA box like we see here, and upstream, such as our CAT box and our GC-rich box. If you mutate most of the bases, what we see is that has virtually no impact on transcription rates. However, when you mutate bases in the promoter or these promoter-proximal elements, that has a huge impact, a huge negative impact on transcription rates. So again, just further illustrating the important role that these DNA regions, these regulatory sequences, play in gene expression. So just sort of thinking about all this schematically here. So here we see a piece of DNA. There's our core promoter with our TATA box. This is the region where various transcription factors will begin to bind. We can identify so-called general transcription factors. These are transcription factors that are essentially required to express any and all genes within eukaryotes. So for example, you might have a TATA box binding protein that binds to the TATA box region, and then various other transcription factors that come into play, and they bind near the promoter or the promoter-proximal element region. When they do that, then facilitates the RNA polymerase, shown here as this purple oval, facilitates RNA polymerase binding to the promoter and eventually making its way down the DNA, transcribing the gene in question. So again, the point is these general transcription factors are required in order for gene expression to take place. In addition to the general transcription factors, by the way, they're often also referred to as basal transcription factors, there are other proteins, other transcription factors that have to be present and must bind to those enhancer regions that we identified previously. So we simply call these specific transcription factors because they are specific to each type of cell or tissue in question. So these would be the proteins that are responsible for triggering the expression of specific genes within specific cells or tissues. And these can serve as activators or repressors. So essentially when they're expressed, they can cause proteins to be produced that again can either stimulate gene expression or inhibit gene expression. So again, just thinking about this in a schematic fashion here. So here's a piece of DNA. Here's our core promoter region. We have our general or our basal transcription factors binding here. RNA polymerase has bound. It's getting ready to make its way down the coding region and transcribe that region. But also notice for that to occur, we must have specific transcription factors binding to enhancers, such as we see downstream here, as well as a few regions upstream here. So again, those they're labeled gene regulatory proteins, but these specific transcription factors, they have to bind to these enhancers in order for all of this to occur here at the promoter, in order for RNA polymerase to transcribe the gene in question. So if you think about it in this way, it perhaps maybe doesn't make sense. I mean, how is it that all of these proteins located way upstream or way downstream, how can they possibly play a role in what's happening here at the promoter region? Well, recall DNA is flexible. It can twist and turn about, and it can loop upon itself. And that is crucial to understanding how proteins binding at enhancers can play a role in assembling transcription factors near the promoter region. So that's illustrated here. So the red structures, they, for example, represent specific transcription factors that bound to the enhancers. That has then, the region of DNA has looped upon itself, facilitating the assembly of all these general transcription factors, shown in gray, which then facilitates binding of RNA polymerase to the promoter, and ultimately leading to transcription of the gene in question. This is just to make the point that, again, if we just go back real quick to the previous slide, you can imagine these red proteins, they must have domains or regions that both bind to specific DNA sequences and also other domains that bind to specific transcription factors. Right? So again, imagine these are proteins, they have three-dimensional shape, but they have two domains. Right? Part of the protein recognizes and binds to DNA, so we say they have a DNA binding domain, and other parts of the protein bind with other proteins, other transcription factors, and we say that's the activation domain. So it's basically the point that these proteins recognize DNA and facilitate the assembly of transcription factors in order to facilitate RNA polymerase doing its job. Toward the left is just simply part of a diagram illustrating in a schematic way that you need both the DNA binding domain as well as the activation domain of these proteins in order for gene expression to take place. Just a phenomenon that we can identify here. It turns out that transcription factors work in a synergistic fashion. So the basic idea we're trying to illustrate here is toward the bottom especially. If you imagine a piece of DNA, here's a core promoter region toward the far right here. If there are no transcription factors present, you're probably not going to have any transcription whatsoever. If you have one of the required transcription factors, well, you might get a very, very small amount of transcription. Okay? It's not necessarily an all or nothing event. And that's sort of illustrated here. So if you have more than one transcription factor, you might have a little more transcription taking place. But this idea of these transcription factors working synergistically, the idea is that the combined effects of all of the required transcription factors is far more than would be assumed by an additive effect, in other words, by each transcription factor working by itself. So the point is when you get all the transcription factors required to express a gene, all of a sudden gene expression just skyrockets, right? So again, the point is these transcription factors essentially work in a synergistic fashion. Now let's take a few minutes and discuss the structure of DNA and how DNA interacts with proteins within cells. So this concept of chromatin. So chromatin refers to DNA that is wound around and tightly interacts with specific proteins, namely histones. So these are illustrated in the figure here. So if we talk about a nucleosome, what we're talking about is this. For example, these 8 purple spheres collectively represents one nucleosome. So what we're talking about here is you have some DNA, close to about 150 or so base pairs, DNA wrapped around all of these 8 different histones, and a single nucleosome contains 2 each of histone 2A, 2B, H3, and H4. Right, so again, that's illustrated here. So collectively, this is one nucleosome. And I just want to point out that if you think about DNA in this state, where it's tightly wound around these histones, and histones are actually tightly connected with each other, that has some very real implications. So first of all, I just want to mention histone H1. Notice on the diagram to the far right, this is a histone that tends to organize the nucleosomes. So again, on the far right, we see several nucleosomes here, and again, all sort of connected and organized via these H1 histone proteins. In this state, where the DNA is tightly wrapped around all the histones, and again, the histones are tightly compacted together, we say that represents heterochromatin. In this state, DNA and the genes within the DNA, the genes are simply not available for transcription. So this tends to be the state of DNA when genes are not expressed. Okay. We can actually see this sort of on a more broad scale. Toward the left, we're looking at an image of some chromosomes that have been stained. You see these dark bands and lighter bands. All those dark regions, that represents heterochromatin. So those are parts of the chromosome, parts of the DNA that are tightly wrapped around histones. And again, those are regions where genes are not expressed. So again, the dark regions are heterochromatin. In contrast, when we have a kind of a loosening or a separation of the histones, much like we see toward this region right here. That is referred to as euchromatin. So what happens is the histones tend to move apart, they relax a little bit, the DNA unwinds a little bit, essentially exposing the promoter regions and the coding regions in order for gene expression to take place. So let's think about that for a few more seconds. And let me just move this. One of the things I want to point out here is this idea of chromatin remodeling. So we're talking about changing the structure of chromatin when genes are to be expressed or not. So in the upper right, we see some histones with some DNA wrapped around it. One of the first things that has to occur in order for a gene to be expressed is this so-called switch SNF protein complex that comes in and essentially nudges the nucleosomes a bit and essentially helps to expose the promoter region of a given gene. In addition, this enzyme, histone acetyltransferase, or HAT, what it does is it adds acetyl groups, shown here as these red dots. Adds acetyl groups to the histone tails. So that's what we see illustrated here. When that happens, that essentially helps these histones relax a little bit and separate from each other. Helps the DNA to relax as well. And again, when that happens, that helps to recruit, for example, the switch SNF protein. So when that occurs, that helps facilitate transcription, or in other words, gene expression. In contrast, another enzyme sort of does the exact opposite. So histone deacetylase, or HDAC, it removes acetyl groups from histones and ultimately leads to lower transcription levels, again because it results in DNA being tightly wrapped around the histones. Another way to visualize this whole process here is shown here. So toward the top we see 4 nucleosomes, each of which is composed of 8 histones. And here we have essentially heterochromatin. So the DNA is tightly wrapped around each of the nucleosomes, again, not being available for gene expression. When those histones are acetylated, that helps to loosen things up and expose the genes for gene expression. When the histones are deacetylated, it goes sort of in the opposite direction. Okay, and again, that's whether they're acetylated or deacetylated. Again, that's due to the activity of the enzymes HAT and HDAC. Okay. Now let's shift gears just a bit and say a few comments about epigenetics. For the most part, when we think about genes, we tend to focus on the sequence of bases found within various genes. And fair enough, that's obviously very important. But it turns out there's a whole other level of control that's involved in gene expression at the epigenetic level. So what we're talking about here are changes to the DNA itself, not to the bases, not to the sequence of bases, but changes that can be made to the DNA, largely, for example, the addition or removal of methyl groups. And so we're talking about changes in gene expression that can occur without a change in the DNA sequence. And some of these epigenetic changes, whether or not DNA is methylated or not, that can be retained through multiple rounds of cell divisions, and it can also be actually passed on through subsequent generations. So again, we're talking about factors that come into play that regulate and have a big impact on gene expression that are really totally separate from the sequence of bases in a particular gene. As an example, this phenomenon referred to as genomic imprinting. Let's take a look and see how this works as an example of an epigenetic phenomenon. It turns out that there are certain genes that have sort of unusual patterns of inheritance. As one example to illustrate how this works, let's think about this situation here, which involves these two genes, IGF2 and H19. You might have a situation where an individual, the cells in an individual that are diploid, so they received one chromosome or one copy of these genes from mom and another from dad. And you might have the sequence of these two genes, the IGF2 and H19, they might be identical, the exact same in the maternal allele and the paternal allele. However, they have radically different expression patterns. How is that? Well, let's take a look here. Initially in the maternal allele toward the top, what we have here is a situation where this protein CTC factor, or CTCF, it can bind to this region right here, this imprinting control region. So this protein CTCF can bind here. When it does so, notice what happens. This enhancer, so again, it's a pretty big protein, right? So this enhancer is simply unable to loop around and bind with this IGF2 gene. So as a result, that gene is not expressed. However, that enhancer can interact with the H19 gene. So the H19 would be expressed. Now let's take a look at what happens in the paternal allele. Again, the key here is that the paternal allele might have the exact same sequence of bases as the maternal allele for both of the genes. But what happens here in the paternal allele is that this ICR region and slightly downstream is methylated. So the DNA is methylated. What are the implications? Well, if that region is methylated, the CTCF protein is unable to bind to the ICR region. As a result, this enhancer, and shown in green here, can interact with and trigger expression of the IGF2 gene. However, the methylation pattern tends to blend over into the promoter region of H19. So as a result, H19 is not able to be expressed. So again, here we have a situation, two alleles, identical sequences from both from the maternal and paternal, but essentially exact opposite expression patterns. Again, all comes down to part of the DNA being methylated, in this case in the paternal allele. So DNA methylation, again, we're not influencing base sequences here, but DNA methylation can have a big impact on gene expression. Let's look at this in an even more broad scale. So think for a few minutes about sex chromosomes in humans and other mammals, right? Think about XX and XY. So males are XY, females are XX. Think about dosage of genes here. If a female has two X's and a male just has one X, you would predict that females would have, whatever genes are on these X chromosomes, females would have twice as many of those genes being expressed as would a male. So what happens to sort of compensate for that potential dosage imbalance problem? Well, it turns out that in females, again, that are XX, during early stages of development, in the cells, one of the two X chromosomes at random are inactivated, and they form a so-called bar body. That's illustrated right here, this little dark structure. So it's basically an inactivated X chromosome. So it turns out that entire chromosome, not just a gene or two, but the entire chromosome is silenced via methylation. So the whole entire chromosome, again, one of the two X's, right, within the cells of a female, one of those chromosomes is entirely silenced, again, via DNA methylation. So that concludes our discussion of gene regulation in eukaryotes.